Okay, I think we'll go ahead and get started. I want to welcome everybody and thank you for joining us today. At the school, as many of you know, the Dean's Lectures are such a special occasion for us, and I certainly value the opportunity to learn more about the work of our distinguished colleagues throughout the school. And that was really the intention of this lecture series, to provide a forum to highlight the extraordinary work of our newly appointed and promoted professors. It is a long-standing tradition and a significant opportunity to celebrate just some of our faculty who make this school so very great. And this is especially true over the last year or so. So thank you so much for joining us, and you will not be disappointed. I'm looking forward to today's lecture. Let me begin by saying that achieving the appointment of professor at the School of Public Health at the university represents incredible accomplishments by a senior faculty member and confirms not only the respect and high regard of the school, but also, and very importantly, of colleagues across the nation and throughout the world. And today's professor is no exception. I am pleased we'll end the academic year with a lecture from our own professor, Adam Spira. Dr. Spira is a professor and vice chair for research and faculty in the Department of Mental Health. He holds a joint appointment in the Department of Psychiatry and Behavioral Sciences at the School of Medicine across the street, and is core faculty of the Johns Hopkins Center on Aging and Health. And I'm going to refer to our speaker as Adam, because I know him very well as Adam instead of Dr. Spira. So Adam's training is in clinical psychology and epidemiology, and his research focuses on the association of sleep disturbance with cognitive and functional decline and neuroimaging outcomes. And he has a particular focus and interest in Alzheimer's disease. He joined the Bloomberg School faculty in 2008 as an assistant professor. And in 2009, he didn't waste any time, and he received a Career Development Award from the National Institute on Aging to investigate the association between sleep disturbance and functional decline in older people. Since then, he has led or co-led several R01 level grants from NIA focused on the association of sleep and/or circadian rhythm disturbance with cognitive decline and neuroimaging outcomes. Again, including Alzheimer's, with a focus on Alzheimer's and including Alzheimer's disease biomarkers. Adam is also a recipient of a SIBAR award. He teamed up with Kristen Kohler and other colleagues to mount a study to investigate how cool roofs improve health equity while at the same time combating climate change. And I hope we'll hear a little bit about the SIBAR award in his presentation today. Dr. Spira, Adam, received a BA in psychology, summa cum laude, from the State University of New York at Stony Brook, and his MA and his PhD degrees in clinical psychology from West Virginia University. He is an elected fellow in the Gerontological Society of America and their section on behavioral and social sciences. He has twice received the Advising, Mentoring, and Teaching Award from our Student Assembly, and has been recognized for his excellence in teaching psychopathology for public health. And last year, he completed the distinguished Johns Hopkins University Leadership Development Program. We are so fortunate to have Adam part of our Bloomberg Schools family. And speaking of family, I would also like to introduce a colleague who needs very little introduction here at the Bloomberg School, Dr. Lainey Rutko, professor of the Department of Health Policy and Management and senior advisor to the president, the university president for National Capital Academic Strategy. Perhaps more famously, at least for today, she is Dr. Spira's significant other. We're so pleased that Dr. Rutko will join us and will facilitate the Q&A session following Adam's talk. So now it is my tremendous pleasure to welcome Professor Adam Spira. Adam, take it away.
Thank you so much, Dean Mackenzie, Ellen, for that very kind introduction and for the beautiful flowers, which I see. Well, I don't know if you can see them, but they are, yes, they're behind me. And I was going, they were going to be repurposed for Mother's Day, but Lainey's on the call, so it's not going to happen. Oh, well. But I'm really pleased to be here. I can't believe it's been roughly 13 years since I joined the faculty, and I'm excited to share with you some of the work that I've been doing, and that I'm very pleased to have been able to do with fantastic colleagues at Hopkins and outside of Hopkins. So I want to start by talking about the aging of the US population. And this is something of which you are likely aware, but it's helpful to put things in perspective to be reminded of this. So if you look here at the figure on the left from the US Census Bureau, you can see that in 2012, there were roughly just over 40 million adults aged 65 and older in the United States. By the year 2050, that will double to over 80 million. And the figure on the right shows this sort of a trend in a slightly different way. You can see the whitish bars indicate the distribution of the population in 2010, and the greenish bars in 2019. And you can see that in just those 9 years, we've lost a chunk of the population that is age 40 to 54, and we've sort of bulked up around the top here, the upper age of the life course. Now, you might be wondering, what does this have to do with sleep? And it turns out that several things, because sleep disturbances are quite common in older people, and poor sleep is increasingly linked to poor later life health outcomes. What sorts of outcomes do I mean? Things that are of critical importance to everyday life for older adults and their family, such as cognitive function, the ability to get things done in one's environment, or functional decline, changes in that regard, and the development of Alzheimer's disease pathology in the brain. This is really critical because late life sleep disturbance is often treatable, and it may therefore be a modifiable risk factor for these key aging health outcomes. So to provide further perspective on why this is important, let's look at the prevalence of sleep complaints among older people. And this is a study of over 9,000 community-dwelling older adults from 3 communities with a mean age of 74, who were asked questions about their sleep. And more than 80% said that they had one of the sleep complaints, either sometimes or most of the time. And when we looked at the authors looked at the proportion of the sample reporting a complaint most of the time, that's what they called a chronic complaint in this study. About 20% had trouble falling asleep most of the time, 30% waking up during the night most of the time. That is, by the way, the most common sleep complaint among older people. 20% had difficulty with waking up too early. A full quarter reported being sufficiently tired that they or sleepy, I should say, that they needed to take a nap during the day. 13% would awaken but not feel rested in the morning. And overall, almost 60% reported one of a chronic sleep-related complaint, one of these or others that they were asked about. So that's a lot of sleep complaints. And the question might arise, well, why are these things happening? Well, some of these, the burden of sleep complaints is attributable to what we believe is normal aging, but a lot of it is not. And I'm going to talk about that now. So this was research done. It was a meta-analysis of sleep parameters, and they collapsed data from across a whole bunch of studies that looked at people all the way from age 5 to age 85. And what they showed was very interesting, that over the life course, we see significant decreases in sleep duration. We see marked increases in WASO, which stands in sleep lingo for wakefulness after sleep onset. So the amount of time that you spend awake after you initially fall asleep. And you can see that this really expands beginning, well, certainly even around 40, it starts to expand pretty significantly. Sleep latency, which is the amount of time it takes you to fall asleep, decreases a little, but not too much. Sleep efficiency, which is the proportion of time in bed that you spend asleep, decreases. So you're spending less time in bed sleeping. And in terms of sleep stages, which you've probably heard something about, we spend more time in light sleep as we get older and less time in our deeper sleep or slow wave sleep. And that has implications for Alzheimer's disease, as I'll discuss later on. There are also reductions in REM sleep, which is the stage of sleep during which we do most of our dreaming. I want you to really pay attention though to the SWS, that's the slow wave sleep, and you can really see that that is curtailed with age. Now, we believe that in healthy people, almost all of these changes have occurred by age 60, and very few of them continue to change in healthy people as we get older. There's a decrease in sleep efficiency beyond age 60. But the problem is that healthy people does not describe the majority of the population, as we well know. So what we believe accounts for a substantial proportion of the sleep complaints that we see in the community are medical and psychiatric conditions and medications taken for those conditions. We think that forms a bulk, that really drives a lot of the sleep complaints that we see. Also, as we get older, there are decreases in physical function that make it harder to do things like stay physically active and get around in the community. And that sort of thing can make it harder to sleep. We know that physical activity and exercise is good for sleep. It promotes sleep. What else? There are psychosocial changes that occur with age that can interfere with sleep and lead to sleep complaints. One of them, perhaps surprisingly, is retirement. We know that people look forward to retirement, perhaps because they'll be able to rest a bit more. But what sometimes happens is, although I must say that many of the retired people I know are busier than I am, it seems, but there is the possibility that one can lose some structure to their day, and as a result, spend more time with napping opportunities. And we have a homeostatic drive to sleep that builds up from the moment we wake up in the morning until we next sleep. And if you nap, you take away some of that sleep drive, so it's harder to fall asleep at night. Retirement is not the only change here. We see that caregiving responsibilities often will begin to place significant demands as one gets older for spouses or for parents, and that can, or just other people in our social networks, and that can lead to sleep disturbances. And then also bereavement is something that unfortunately, as time goes on, we're faced with more of. So those are other contributors. You may well have heard of circadian rhythms. These are the many physiological processes follow a circadian rhythm, or an approximately 24-hour rhythm. And that's those processes, including things like core body temperature fluctuations and hormone production and release, are under control of the master clock in the brain, the suprachiasmatic nucleus, or SCN. And the sleep-wake cycle, the daily alternation of sleep and wake, is really one of the clearest manifestations of the circadian rhythms, and in concert with that sleep homeostat that I talked about. So in older adults, there is a circadian phase advance such that older adults tend to get sleepy and go to bed earlier and wake up earlier than younger people. And that can lead to sleep complaints when, for example, a person might complain, well, I'm only sleeping until 4 a.m. Well, if one is going to sleep at 9 p.m., that's a solid chunk of sleep. If one wakes up at 4 a.m. but is going to sleep at 9 p.m. So that can lead to sleep complaints, even if sleep is of sufficient duration. But a very important thing to consider is that there are, aside from all of the things I already mentioned, sleep disorders that can affect one's sleep and one's perception of one's sleep, such as sleep disordered breathing, which is very common in older adults, as I'll explain. So Dean McKenzie was very kind to refer to my career development award that I received when I, in 2009, after coming to Hopkins. And in that grant, we were really focused on examining links between poor sleep with functional decline and disability in community dwelling older people. And I want to share some of the findings from that research with you. So this was a study that I published with colleagues, including Dr. George Reebok, who was a fantastic primary mentor on my K Award and is today a terrific colleague, collaborator, and friend, and with mentors at UCSF, including Ken Kavinsky and Christine Yaffe, and elsewhere. And we did this study because even though there was preliminary evidence that linked poor sleep with functional limitations and disability in older adults, most studies were cross-sectional and most relied on self-report measures of sleep. And it turns out people are not, in general, very good historians regarding their sleep, even the sleep of the last night, let alone the last 7 nights. So what sleep researchers have done for decades now is rely upon accelerometers mounted on the wrist, which we tend to call wrist actigraphs, to quantify movement. And we have algorithms that have been derived from studies that have validated them against polysomnography. That's the full head to toe sleep study that is really the gold standard sleep measure. And we apply those algorithms to the activity data from these devices, and we get estimates of sleep duration and sleep fragmentation, sleep efficiency, et cetera. So most studies there weren't really hardly any studies that had done that. And neither were there studies that had used objective measures of functioning. So we asked in the following question: in community dwelling older women, what is the association of objectively measured sleep with incident instrumental activity of daily living, or IADL difficulties, and decline in performance-based measures of physical function? And we did this research in 817 women in the study of osteoporotic fractures, or SOF, which is a prospective study of aging that you'll hear a lot about in this talk. Now, this study recruited almost 10,000, mostly white women, in 1986. They recruited white women because it was a study originally of the causes and consequences of osteoporotic fractures, which are more prevalent in white women. The inclusion criteria for that study were age 65 and older, ability to ambulate independently, and absence of bilateral hip replacement. And fortunately, in 1997 and 98, the investigators added 662 Black women to the cohort to add some diversity. Now, these participants completed in year 16 of SOF, they completed wrist actigraphy for at least 3 24-hour periods using a medical grade Fitbit device like those accelerometers that I've showed you. Happy to answer questions about consumer grade wearables if you like. But and then at year 16 and year 20, they completed measures of functioning, including self-reported IADL difficulty, so difficulty with grocery shopping, heavy housework, and preparing meals, and objectively measured performance, including grip strength and gait speed. And you can see some of their characteristics here. They were age 82 on average, about 13% non-white, 40% had beyond a high school education. And here you can see some of their actigraphic sleep parameters broken down, in this case, by quartiles. I'll give you the averages. On average, their total sleep time, which is the total amount of time spent asleep while in bed, was 409 minutes, which is 6.8 hours. Their wake after sleep onset, or WASO, the amount of time they spent awake after they initially fell asleep, was 66 minutes. And their sleep efficiency, the proportion of time in bed spent asleep, was 80%. So of these 817 women we studied, 41% had incident difficulty in at least one IADL at follow-up about 5 years later. You can see the breakdown here. And I'll start by saying there was no association between sleep parameters and gait speed. So what did we find? Well, after accounting for a range of potential confounders, which you see at the bottom here, we found that women in the quartile with the shortest sleep duration had double the odds of incident IADL difficulty compared to those in the quartile with the longest sleep duration. We also found that, well, first of all, I should say there was a wake after sleep onset was interestingly not significantly associated with incident IADL difficulty. But we did find that compared to women who were in the highest quartile of sleep efficiency, those who spent the most time in bed asleep, the highest proportion of time in bed asleep, those compared to them, those who were in the two lower quartiles, that's actually the upper quartiles, but those are the two that have the least amount of time in bed spent asleep, had about 1.7 times the odds of incident IADL difficulty. And then when we looked at decline in grip strength, which we defined as being in the highest quartile of decline in grip strength, which was more than 4.75 kilograms of weakening in hand strength over 5 years, we found that compared to those who had the longest sleep duration, those who had the shortest had about double the odds of significant weakening in their grip strength. And compared to those who had the least wake after sleep onset, those in the quartiles with the most had 1.9 times the odds of declining grip strength. And then sleep efficiency, we saw those with the worst sleep efficiency compared to those with the best had double the odds of a significant decline in grip strength. So we wanted to follow up this study to say, well, that's interesting. We can see that in the community, these folks, their ability to take care of themselves changed. What are the implications for nursing home placement? So we did another study using the same cohort and with similar authorship, you can see. And we used data from soft year 16 again, when they did the actigraphy. They provided information about their place of residence. They also provided that at year 20. And we studied people who were community dwelling at that year 16 baseline. So that meant that they resided in a private home or apartment or a retirement home / senior complex. And we looked at associations with placement in a nursing home at follow-up. And you can see 5 years later, of the 1,664 women in the study, about 4.3% were in a nursing home at follow-up and 7.6% in an assisted living facility or ALF at follow-up. Again, we found no association between total sleep time in either nursing home or assisted living facility placement. However, we found that compared to the women who had the least wakefulness after sleep onset, those who had the most had 3 times the odds of being in a nursing home 5 years later. And that's after accounting for all of these potential confounders. And you'll see here the P for linear trend indicates that there is some evidence of a linear increase in the odds of our outcome across quartiles. We also found that, and this was very interesting as well, that 2.5 to, well, those who were in the lowest quartiles of sleep efficiency, who spent the smallest proportion of time in bed asleep, had 2.5 to 3.25 times the odds of being in a nursing home 5 years later, compared to those who had the best sleep efficiency. And similarly, we found that those who had who were the 2 highest quartiles of wakefulness after sleep onset had about more than twice the odds of being placed in an assisted living facility 5 years later. And again, we saw that having poor sleep efficiency was associated with more than double the odds of being placed in an assisted living facility. So just to summarize these findings, there were some of the results from my K award. it looks like objective indices of disturbed sleep are indeed associated with an increased risk of incident IADL difficulties and substantial decline in grip strength, as well as nursing home and assisted living facility placement. But of course, one of the questions that comes up is, well, what are the mechanisms that might be linking poor sleep to these adverse outcomes? One possibility, even though we attempted to adjust for medical comorbidities, there may have been some that were unaccounted for. So there may have been residual confounding. There may also, one of the potential, we know that sleep disturbance promotes inflammation, and inflammation is bad for when it's chronic for all sorts of functions and organ systems. So it could be that that played a mediating role. It could also have been that there were subtle preclinical neurological changes that weren't necessarily measured by the cognitive tests that we might have adjusted for, that might have driven both the poor sleep and the decline that necessitated nursing home placement, et cetera. So regardless, this was pretty intriguing data from my perspective, and it suggested that maybe there's a role for sleep and sleep optimization in the prevention of disability and nursing home placement in older people. Now, while I was doing this work, I became increasingly interested in links of disturbed sleep with the cognitive decline and neurological changes that that characterize dementia. And I want to talk a bit about that now. So about 14% of adults age 71 and older, and almost 40% of those 90 and older, have dementia. And Alzheimer's disease, or AD for short, is the most common cause. Two hallmarks neuropathologically of Alzheimer's disease are extracellular beta-amyloid, or A-beta plaques, early in the disease course, and intraneuronal, or within the brain cell, neurofibrillary tangles later in the disease course. And those tangles are associated with brain atrophy and loss of brain cell death and atrophy. In 2010, there were almost 5 million adults age 65 with AD in the US, but there will be 14 million in the US by 2050. And this is not just a local problem. Globally, AD prevalence will quadruple between 2000 and 2050. There is no cure for Alzheimer's disease and other forms of dementia, so prevention is critical. So in the past, conventionally speaking, when we would hear researchers or clinicians talk about neurodegenerative disease and sleep, this is the framework that was typically invoked, that neurodegenerative disease was causing, and this disease that causes Alzheimer's disease, dementia of the Alzheimer's type or other forms of dementia, the changes in the brain were driving sleep-wake disturbance by changing parts of the brain that modulate sleep and wake. And a lot of work mostly in the 1980s and 1990s focused on this, comparing the sleep of people with Alzheimer's disease to cognitively normal older people. And what they found, by and large, is that in Alzheimer's disease and related dementias, we see an exaggeration of the aging-related changes that I described as normal. These include, as you might remember, shorter and more fragmented sleep. This is in people with Alzheimer's disease compared to those without. Lower sleep efficiency and reduced slow-wave sleep and REM sleep. And interestingly, greater dementia severity was linked to greater sleep fragmentation in folks with AD. The animal literature bore this out as well. Rowe et al. From David Holtzman's group at WashU in St. Louis showed in an Alzheimer's disease mouse model that as amyloid plaques accumulate in the brain, the sleep-wake cycle is altered. And they showed that active immunization of these AD mice with A-beta 42 peptide prevented the sleep-wake cycle alterations. So yes, indeed, changes in the brain are contributing to the sleep-wake disturbance that we see. So some of those, one of the brain mechanisms is loss of volume, we believe, in the suprachiasmatic nucleus. Remember, that's the clock, the master clock in the brain, in the anterior hypothalamus, which is the synchronizer of all the clocks in all of the cells of our body, and that's what keeps us in tune with the environment, the dark light cycle. And also in the intermediate nucleus, there's brain volume loss. This is a figure showing suprachiasmatic nucleus volume in people of various age groups, and of interest here is a comparison of folks with dementia to older adults without dementia, and you see that there is a decrease in that group. But environmental factors are very important as well. So we know that people who have dementia, particularly if they're living in a nursing facility, tend to have reduced exposure to bright light. And bright light is the primary Zeitgeber, or time giver in German. And that is what we refer to as environmental cues that help entrain the circadian system. So reduced exposure to that bright light can lead to disruptions in the sleep-wake cycle. Also, reduced daytime activity and more napping can make nighttime sleep, can worsen nighttime sleep. And alterations in the cholinergic system, which we know occur in Alzheimer's disease, are believed to affect sleep as well. So plenty of reasons to believe that neurodegenerative disease affects sleep and wake. But there was a seminal study published in 2009 that started to get us to scratch our heads a bit. And namely, that was by Kang et al. From Dave Holtzman's group again in WashU. And what they showed is that when they looked at Alzheimer's disease mice, if they sleep deprived the mice or restricted their sleep significantly, they overproduced A beta protein and ended up with much greater plaque burden. So sleep deprived AD mice, you get more plaque in the brain than if you don't. The white bars are the controls, the black bars are the sleep restricted mice. And my colleague, Mark Wu in the School of Medicine, with whom I've since collaborated on a lot of stuff in this domain, he and I got together and we were awarded a grant from the Johns Hopkins Brain Sciences Institute back in 2010, I believe, to look at links between sleep disturbance and amyloid deposition, one of the hallmarks of Alzheimer's disease, both in humans and in fruit flies, in Drosophila. And what you see on the right side is results from the fruit fly research led by Masashi Tabuchi. And you can see here that, so what you have to know about fruit flies is they sleep at night like humans. And this is a fly model of Alzheimer's disease. And if you shake the tubes in which these fruit flies live at night, you get this amount of amyloid deposition. This is an immunostain slide in the fly central nervous system. If you shake it during the day when they're awake anyway, you get much less. So these studies and studies like them began to make some compelling arguments for a causal link of sleep deprivation, sleep disturbance, insufficient sleep as a factor in the etiology of Alzheimer's disease. Now remember, that was just for A beta deposition. Tau protein is what forms the neurofibrillary tangles once it's hyperphosphorylated. And what we've seen more recently, also from Dave Holtzman's group, is that if you sleep deprive a wild type mouse, you actually increase tau in brain interstitial fluid. So both A beta and tau are affected by sleep deprivation. And this was a bit of a sea change, and people really started to pay attention to sleep-wake disturbance as a factor that may be driving neurodegenerative disease. And my colleagues and I took notice, and we started to do more and more research in this area. I'm going to show you some of that work now. We did a study that we published in 2013. This was a terrific collaboration with investigators from the National Institute on Aging Intramural Research Program, led, you can see Susan Resnick is the senior author here, along with Dr. Luigi Ferrucci, the scientific director for NIA, and along with folks at Johns Hopkins, namely Mark Wu, we did this study of self-reported sleep and amyloid deposition in 70 Baltimore longitudinal study of aging, or BLSA, participants with a mean age of 76. And you can see they were about half women, 20% African American, 17 years of education on average. This is a very highly educated cohort. The MMSE is the mini mental state exam, and it has a top score of 30. These folks were in pretty good shape, as you can see. And they, as I mentioned, self-reported their sleep, and within 5 years had an amyloid scan by PET, which enables us to quantify and actually see amyloid plaques in the brains of living humans. And so what we found was shorter sleep duration, reports of shorter sleep duration were associated with greater amyloid deposition according to a cortical DVR, distribution volume ratio, which is a continuous measure of plaque burden and it's the cortical one is sort of a global measure throughout the brain. And we also saw that it was associated with amyloid deposition in the precuneus, which is a region that's affected early in the AD course by amyloid deposition. We didn't see in this cohort, we saw a statistical trend towards an association in the expected direction for trouble falling asleep. We didn't see any association with respect to reports of wakefulness during the night, but we found that reports of poorer sleep quality, less restful sleep, were associated with greater amyloid burden. And the figure on the right shows you these are, well, really the unadjusted association of sleep duration with plaque burden, amyloid plaque burden. These are different strata or slices of the brain. And this column on the left, it's hard to appreciate. These are the brains of people who reported sleeping more than 7 hours. And then we have folks who report getting more than 6 to 7 or fewer hours in the middle. And on the right here, the third colorful column of brains represents folks who reported 6 hours or fewer of sleep. And you can see as you move from left to right, from longer to shorter sleep duration, you get hotter colors, which indicate shorter sleep duration is linked to greater amyloid deposition. In another study that we published in the journal Sleep, we took advantage of the fact that prior to those amyloid scans, a good number of people in this cohort had reported how, they'd answered an important question about excessive daytime sleepiness. And I decided that, it would be a great idea to leverage that for a study. So they were asked, do you often become drowsy or fall asleep during the daytime when you wish to be awake, e.g. falling asleep watching TV or reading? And what we found, surprisingly, with a single yes/no question, is those who report, compared to those who were negative for excessive daytime sleepiness, those who responded yes to this EDS question had about 2.8 times the odds of having an amyloid positive scan. So having significant amyloid deposition on a PET scan, an average of 16 years later. So that was quite surprising. And it really got us thinking. There are lots of different things that can lead to excessive daytime sleepiness, including medical comorbidities, including other sleep disorders, which I'll talk about in a moment, and chronic insufficient sleep. So, and that's something that we still haven't answered the question exactly yet. But what I can tell you is we know that at least one of the sleep disorders I'm talking about before, I mentioned before, sleep disordered breathing, is associated with bad brain health outcomes. So that might be what we're seeing. And I want to talk about that briefly. So sleep disordered breathing is characterized by repeated interruptions in respiration during sleep, cessations of breathing, which are referred to as apneas, apnea without breath, and significant reductions in airflow associated with blood oxygen desaturation, which are referred to as hypopneas. Now, the most common type of sleep disordered breathing is obstructive sleep apnea, or OSA, as it's abbreviated. The risk factors for which are age, older being associated with a greater risk, greater BMI, body mass index and a greater neck circumference, and being male is associated with a greater risk of sleep disordered breathing. Now, sleep disordered breathing is measured by polysomnography, that full head to toe sleep study that I talked about before, which is the gold standard for sleep measurement. And one of the indices that is commonly used to quantify the severity of sleep disordered breathing is the apnea-hypopnea index, which is the number of apneas plus hypopneas per hour of sleep. And SDB, sleep disordered breathing, is highly prevalent in older people. Depending on which study you look at, you can get a wide range, but one study here found roughly 20% of people aged 60 to 99, and another found as many as 62% of adults 65 and older. I think that there's no, while that's a wide range, there's no doubt, I think at this point, that there's a substantial population burden of sleep disordered breathing. And I was a co-author on a study led by Christine Yaffe at UCSF in the soft cohort, looking at 298 women with a mean age of 82 who in SOF who had a median apnea-hypopnea index, or AHI, of 10. And of all of those women, 35% had moderate sleep disordered breathing as defined by an AHI of 15 or greater. And what we found was that compared to those without sleep disordered breathing, per this definition, those with had 1.9 times the odds of having subsequent mild cognitive impairment, or MCI, or dementia diagnosis 5 years later. And we saw that the number of oxygen desaturations per hour was strongly associated with the odds of MCI or dementia, as was the proportion of sleep time spent in apnea or hypopnea. So it seems like the hypoxia is, guess what? Turns out it's not good for your brain. And that's important because of the population burden of sleep disordered breathing. Now, I want to get back to some neuroimaging research that I did. I showed you some A beta deposition findings, and now I want to show you some results looking at brain volume loss, specifically looking at links between sleep duration and subsequent cortical thinning in cognitively normal older adults in the Baltimore Longitudinal Study of Aging Neuroimaging Initiative. So we studied 122 individuals who were 67 on average when they reported their average sleep duration, and they subsequently completed, on average, about 8 MRI scans. And we used repeated measures of cortical thickness as our metric of gray matter atrophy. And we only used data for this study from visits at which participants were cognitively normal. So the data you'll see here are all from cognitively normal people. Because of the distribution of sleep duration in this sample, we categorized sleep duration by less than 7 hours, 7 hours, and greater than 7 hours. And compared to people who reported getting 7 hours of sleep, those who reported fewer than 7 hours had more rapid cortical thinning over the subsequent period in temporal and frontal brain regions, almost all of which were in the left hemisphere for some reason. We're not exactly sure why. And interestingly, compared to those who had an intermediate sleep duration, if you will, of 7 hours, those who reported more than 7 hours of sleep on average had more rapid cortical thinning in frontal regions. And I'm happy to chat later about why that might be the case, that more sleep is not necessarily associated with better brain health outcomes. And here you see some pictures of the associations I just described, comparing people who reported fewer than 7 hours to 7 hours and more than 7 hours to 7 hours. Now, as with the functional decline data I showed you, a big question that comes up is, well, how is this happening? What is the mechanism by which poor sleep might be leading to Alzheimer's disease pathology or brain volume loss? And Yoel Ju and colleagues from WashU published a paper that I think gave a very nice figure illustrating what we think is going on, which is that poor sleep quality is associated with lower levels of slow wave sleep. And this is, remember, we see in older adults that on average, you're going to get markedly less slow wave sleep than you did when you were younger. But we think that if you have poorer sleep quality, it's going to further degrade the amount of slow wave sleep you have. And that's important because we think that slow wave sleep plays a key role in offsetting some of the changes in our brain that occur from the time that we wake up until the time we go to sleep and enter slow wave sleep again. Namely, as we go through the day and we interact with the environment, we learn things, etc. There are lots of connections that are made between our neurons, and those are good things. They are required for us to, to learn. But there needs to be some downscaling of those connections and that activity. We think that usually of brains, having brain activity as being a good thing, and yes, it is. But we think the key role that slow wave sleep may play is that it sort of, there's all of this intensity and activity that's built up that needs to be reset. It's almost like a reboot while we sleep. And it turns, well, I was going to talk about how implications for long-term memory and things like that, but we don't have enough time. It's a fascinating set of concepts. But excess neuronal activity prompts A beta release. And so without the slow wave sleep checking that neuronal activity, we get excess A beta. And then that leads to more A beta in brain interstitial fluid and an increased risk of plaque formation. But the story doesn't end there. The plaque formation eventually, we think, and other AD-related processes lead to symptomatic Alzheimer's disease. But we think that what happens is the plaque also forms in brain regions that control sleep, as I showed you I talked about earlier, and that further degrades sleep quality, leading to a vicious cycle. Now, I also want to mention sleep disordered breathing, which does two things. It fragments your sleep, degrading your sleep quality, and thereby going through this whole, so it could lead to A beta through this mechanism and dementia through that mechanism, but also it leads to hypoxemia and inflammation, which are not good for the brain and are known to play an important role in the production of amyloid plaque. So that's another mechanism by which sleep disordered breathing could enhance Alzheimer's risk. In 2013, there was a fascinating paper published in the journal Science called Sleep Drives Metabolite Clearance from the Adult Brain by Xi et al. In Megan Nadergaard's group at University of Rochester. And they showed in mice that slow wave sleep increased the clearance of A beta, exogenous A beta that was injected into the mouse brain. So inject this A beta into the mouse brain, and if they sleep, or interestingly, if they're anesthetized, they will have an increased clearance of exogenous A beta, and it only happened during slow wave sleep, interestingly. And the mechanism by which this happened, what has been referred to as a brain dishwasher effect, is an expansion of the interstitial space. There is, as it says here, natural sleep or anesthesia were associated with a 60% increase in the interstitial space, which then facilitated exchange of brain interstitial fluid with cerebrospinal fluid that ended up flushing out the A beta. Now, this is mediated by aquaporin-4 water channels in astrocytes, which are a type of glia, which are non-neuronal brain cells. And if you take the lymphatic system is responsible for clearing toxins throughout the periphery. Well, they added the gla for glia and called it the glymphatic system. And exciting research is happening in this area. It's been in the press a lot, and further research is needed to show what the implications are for Alzheimer's disease in humans, but we could say more about that. But in summary, self-reported shorter sleep, poorer sleep quality, and excessive daytime sleepiness are associated with A beta deposition on amyloid scans. Shorter and longer sleep reports of those things versus intermediate sleep duration are associated with higher rates of subsequent cortical thinning in frontal and temporal regions, and animal studies indicate a bidirectional link between disturbed sleep and A beta deposition, and that sleep deprivation increases tau. So it really looks like there is a bidirectional link here between neurodegenerative disease and sleep-wake disturbance. but many questions remain unanswered, such as how does sleep disturbance interact with things like sex, age, and genetic factors to affect AD pathophysiology and other aging-related outcomes? And how we know that actually there's a large vascular contribution to dementia and to Alzheimer's disease, which was not always appreciated. We know that there are that sleep disordered breathing and short sleep can increase cardiovascular disease risk. How does sleep interact with vascular risk factors and disease to affect brain health? We need to know. What is the role of circadian rhythms or the timing of sleep or sleep-wake patterns in these associations? Much more to study there. And then the big question is, does treatment of insomnia or the optimization of sleep aid the prevention of cognitive and functional decline in Alzheimer's disease? So to answer the question, is good sleep the keystone of healthy aging? Well, if it's not a keystone, I would extend the architectural metaphor.
I'm learning how to choose the right audio apps.
Okay, Siri was chiming in with the answer apparently. If it's not a keystone, it's a pillar, I would say. But I want to go broader here to ask, is good sleep the keystone of public health? And before I answer that, I want to show you something about sleep duration in the US population. And please focus on the orange and red in this figure. This is a great book by Michael Grandner called Sleep and Health, by the way, if you don't have it yet. But 37% of the US population of adults get 6 hours or fewer of sleep. And that is a problem. Why? Well, this is an infographic that was published by Hopkins Medicine. And you can see here, this has implications for so much of public health. This is the effects of sleep deprivation. There are effects on safety. 6,000 fatal car crashes caused by drowsy driving each year. We have, when we are sleep deprived, we have more cravings for sweet, salty, and starchy food. It changes our hunger hormones and our appetite hormones. It modulates those, and it turns out that you're 50% higher risk for obesity if you get fewer than 5 hours of sleep per night. It affects immunity such that it increases our risk for colorectal cancer by 36%. It increases our risk of high blood pressure and stroke. It increases the risk of heart disease, and you're 3 times more likely to catch a cold. This may have implications for COVID, which, by the way, has really destroyed our sleep. That's a whole other conversation. And you see the dementia risk here. And sleep deprivation can age your brain by 3 to 5 years. So those are very important things. And actually, a 2006 IOM report was entitled Sleep Disorders and Sleep Deprivation, an Unmet Public Health Problem. And this is a quote from Janet Croft from CDC. Sleep and sleep disorders are increasingly recognized as vital to a wide variety of public health and chronic disease concerns, including obesity, hypertension, and cancer. And then breaking news here from Adam Spira, May 5th, 2021. When we neglect sleep health, we undermine public health. So there you have it. At least 2 out of 3 sources were authoritative. Now, I want to talk about something serious, something else serious here, which is sleep disparities and the effects of sleep disparities potentially on health disparities. If you look at this figure, you can see from using data from the National Health Interview Survey that goes from 1977 through 2009, that there, if you look at the sleep of white folks compared to the sleep of Black folks, those who get 7 to 8 hours sleep, you can see there's a big gap with white people getting a substantial proportion of white people, substantially greater proportion of white people getting 7 to 8 hours of sleep, which is a good solid amount of sleep in the US compared to Black people. And we talk a lot about health disparities, and I believe that sleep disparities may very well be a driving force here in some of the health disparities that we study. I've done a little bit of work in this myself with trainees, Chris Kaufman and Becky Hawk and colleagues, in the Department of Mental Health. I've been interested in neighborhoods and neighborhood characteristics, which we know can certainly, well, I'll talk more about neighborhood in a moment, but and the effects that they can have on sleep health. And people this is really becoming, there's a groundswell of research in this area. You can see a study here, senior author Chandra Jackson at NIEHS is doing terrific work on sleep disparities and sleep health more broadly. But what is the role of race and ethnic discrimination on, for degradation of sleep health? Really important work. Now, along these lines, Dean McKenzie had said she hoped that I talk about SciBar, and I'm thrilled that Kirsten Kohler, Associate Professor in EHE, and I received funding from the Bloomberg School of Public Health SciBar initiative to conduct research on the effects of climate change on the urban heat island, which reflects the disparities in heat between urban centers and rural or suburban areas, and disproportionately affects lower income and minority communities. In this study, we'll be examining the effects of cool roofs, lightly colored roofs that reflect sunlight rather than absorbing it, as shown in the picture here with one of our co-investigators, Emmanuel Drabo. And specifically, we'll be examining the effects of these cool roofs during the summer months on bedroom temperatures, indoor air quality, and sleep, which is negatively affected by high temperatures, and a range of other health outcomes in Baltimore City residents. And the hope is that we'll show that cool roofs reduce indoor temperatures in the summer months and subsequently improve health, including sleep. And this would point to cool roofs as a means of improving health and health equity in Baltimore and other cities. And finally, I'm gonna say thank you. I didn't leave much time for questions. Thank you to the NIA who has funded so much of my research. I'm very grateful. I wanna thank Dean McKenzie and my Department of Mental Health colleagues. And I'm so lucky to have such terrific people to work with in the department and the school and outside of Hopkins. And my co-authors and collaborators, and my trainees who make this such fun, including Darlene Rojo-Wissar for her help with slides, some at the last minute. Thank you, Darlene. And the participants in the study of osteoporotic fractures and BLSA, and the people depicted here, these characters are my technical, emotional, and life support team. So thank you, thank you, thank you.
Adam, I think if we do it as a speed round, we can get through a couple of questions.
Terrific.
Okay, so first question. What are your thoughts or existing research about aging physicians and the 24+ hour call schedule? Is it bad for the provider and or the patient? Instead of shaking the fruit fly test tube, how about ringing the pager bell? I wish I didn't add that last part. That is from the questioner.
That's a good one. I like that. So I think that there are certainly implications here for a lot of different aspects of a lot of different occupations. Occupational health, I think, and work performance actually has a lot to do with how we sleep, but our jobs can affect how we sleep as well. So shift work, for example, is linked to a bunch of bad outcomes, including cardiovascular consequences. And I wonder how much that is due to sleep loss, for example. Certainly an important point that was raised about aging physicians and sleep, and not just aging physicians. Something that's important to note is we don't know if there's a critical period during which poor sleep has a really adverse effect on brain health, like midlife hypertension does for Alzheimer's disease. Okay. Anything else?
I'm going to try to sneak in one more. Is it possible to reverse the effects of consistent self-imposed, meaning not health-related, minimal sleep, so under 6 hours a night, just by sleeping more? How do we know how long it takes to get our bodies back on track?
There is a whole literature that looks at recovery, and I can tell you that it is not as fast as we would like. There are people who have done a lot of this research, particularly at the University of Pennsylvania, and it is not as simple. I wish it were simpler, and it takes a longer time than we would like. And you ask about how it affects our bodies, but I think it could affect many different parts of our bodies differently. So I can't really answer that, but good question.
Well, I take this job very seriously, and it's 5 p.m. and I want to make sure that we wrap up on time.
Let me just, oh, go ahead, Lainey.
Oh, can I sneak in? I see we just got it. Can I sneak in one more question, Ellen, or are we?
Yep.
Okay, one more. You gave compelling evidence that sleep may have a causal role in dementia. Are you planning to engage in sleep intervention or implementation research as a next step?
Yes. And I think that we've gotten to the point where there's sufficient epidemiologic evidence, and I didn't even have a chance to present. There are studies in healthy young people showing that there are changes in cerebrospinal fluid dynamics of A beta, even with a single night of sleep deprivation. So I think that we're getting to the point where drug trials are going to be happening. It's a whole other question. Who do you select for that study, and how do you study? A lot of questions there, but yes, that is the answer.
Great. All right. So maybe your son has a question. No?
Alex, do you have a question?
Do you have a question for your dad?
It's just a wave. I'll take it.
Well, that was terrific, Adam. And there are a number of comments in the chat saying, terrific presentation, such an interesting topic, and I think a very appreciated topic in public health. So we're all thrilled that you're tackling this. And as an aging person who has some sleep disturbances, I hope you find the answer soon. But
Thank you, Ellen.
it's great work, important work, and we're so honored to have you on our faculty. Keep up the good work. And thank all of you for joining us this afternoon. And if you have any further questions for Adam, I'm sure he'd be more than happy to engage with you over email.
Absolutely. Email me.
And Lainey, thanks for your terrific moderating.
Yeah. Thanks, sweetie.
All right, guys. Take care.
Thanks.
Bye-bye.